Hello everybody, today we are going to solve October-November 2018, Paper 6 Variant 3, Biology. The code of the subject is 0610, and we're going to solve Paper 6 Alternative to Practical. Let's begin. Question number 1. Catalase is an enzyme found in plants and animal cells. It catalyzes the breakdown of hydrogen peroxide to form water and oxygen. As we see, we have 2H2O2, which is hydrogen peroxide, reacts to make 2H2O plus O2. 2H2O, which is water, O2, which is oxygen. The oxygen produced during the reaction forms a foam on the surface of the mixture of hydrogen peroxide and catalase. The height of the foam can be used to measure of the activity of the catalase present. As we see right here, a student investigated the effect of cooking on the activity of catalase. That means we have the independent variable that we're going to use later on in other questions. In the potato tissue using 3% of hydrogen peroxide. Now the student used the following method. Let's read the steps. Step 1. Cut 2 potatoes so that they are identical in both shape and size. So this is a constant variable. Now step 2, put 5 centimeter cubic of hydrogen peroxide solution into a test tube labeled uncooked potato. But right here in step 3, put 5 centimeter cubic of hydrogen peroxide solution into a test tube labeled cooked potato. In step 4, we put one of the potato sticks into a beaker of water, 90 Celsius. That means that the enzymes in the potato stick denatured, and we leaved it for 5 minutes before carefully removing it. 5 minutes is the constant variable. Now step 5, put the potato stick from step 4 into the test tube labeled cooked potato. In step 6, put the uncooked potato into the test tube labeled uncooked. Step 7, leave the potato sticks in the hydrogen peroxide for 3 minutes and then measure the height of the foam produced in each of the test tubes. So we're going to measure at the end. Next question. Figure 1.1 shows the test tubes after 3 minutes. We have the uncooked potato and the cooked potato. Here we have the foam produced by the catalase reaction, and here we have the potato sticks in each of the test tubes. Let's go to question AI. Prepare a table for the student's results. Measure the height of foam in each of the test tubes in figure 1.1. Record these values in your table. Before we begin, when we draw a table, we need to use a pencil and a ruler to have higher accuracy. So I'm going to now get a pencil and a ruler. Let's begin. So measure the height of foam in each of the test tubes. So we're going to outline a table like this. Neatly, as I said. Okay. And then I'm going to divide it into 2 parts. The first part is the test tubes. So test tubes. And in the second part, I'm going to write height of foam. And of course, I need to include the units, which is per centimeter. Now, I'm going to write down the cooked and uncooked potato results. We have the cooked potato, which had a height of foam of 9.6 centimeters, and the uncooked potato had a height of 11.8 centimeters. Right here, we don't have to include the units because we have included it up here in the title. Now, question II. Calculate the difference in the height of the foam produced by cooked and uncooked potato after 3 minutes. Include the units. Now this question only has 1 mark and we only have 1 step to do, which is to subtract 11.8 minus 9.6, which is equal to 2.2 centimeters. Write that down. Let's go to the next question. Describe how you made sure that your measurements of the height of the foam in each test tube could be compared. Now, the answer is to avoid parallax, of course. To avoid parallax error by looking at the test tube perpendicular and measure from the same point. So in this way, we are going to avoid error, decrease the positive false. In this way, we can find the animal's result more easily. Now, in question IV, it says state a conclusion for these results. So basically, the conclusion is that the cooking potato, or the cooked potato, decreases the catalase activity. And it produces less oxygen gas or foam, which is one of the products of the catalase reaction. Let's go to the next page. Now in question BI, it says identify the variable that was changed or the independent variable in this investigation. So we have many independent variables. The most important one is the treatment of the potato. It's how the potato was treated. Now, let's write down the treatment of the potato. If cooked or uncooked. Okay. Now, in double I, the question says, state 3 variables other than the way in which you measured the height of foam that should be kept constant in the investigation. Constant variables are the variables that are not changed to have a fair test. So one of the main constant variables is the volume of hydrogen peroxide. So volume of hydrogen peroxide. Also, the size of the potato is the same as we read earlier in this video. So let's write down the size of the potato or potato stick. Okay, and the third constant variable was the time in the peroxide solution, which was 5 minutes. Let's write it down. Time in the peroxide or the hydrogen peroxide solution, which was 5 minutes. Now, in question C, the question says, identify 3 sources of error in this method. So, one of the errors was that the experiment wasn't repeated. And it's very important to repeat an investigation in biology to get more accurate results, to divide and get the average to identify the anomalous results. Now, the second point, it's the height of the foam was difficult to measure. So let's write it down. The height of the foam was difficult to measure. Also, it was difficult to measure because the bubbles or the bubble height wasn't even, it was uneven. So I'm going to add uneven height. This will decrease the accuracy of the investigation. Now, point number 3, that one potato was soaked in water and the other wasn't. So let's write it down. One potato was soaked in water and the other wasn't. Now let's go to the other page. Here we have question D. Question D has 6 marks, as we see. Now the catalase student was asked to the hypothesis. Catalase activity is the same in all species of plants. This is the hypothesis of the student. Now plan an investigation to test this hypothesis. Now we're going to plan an investigation, but I want to list that we have many things to write down while planning an investigation. We have the independent variable, the dependent variable, the control variable, the time, and the tools, safety, and of course we have to repeat the investigation at least 3 times to get accurate results. Now let's write it down together. The first point, to get 5 different plants. I'm going to list 5 different plants. It could be any 5 random different plants. Let's say potato, tomato, cucumber. We have mint, and let's add guava. Okay. Now the second thing, we must obtain an extract from them, which is the plants, by crushing. Now we must list the name of the tool we use it for crushing, because we must list the tools in investigations. We must grind by mortar and pestle. Mortar and pestle. And then we add into each test tube the same volume and same concentration of extract and of course hydrogen peroxide. Also, I want to add that we must have the same temperature, which is one of our constant variables. Of course, how could we obtain the same temperature or keep it constant? By using a thermostatic controlled water bath. And we have one more constant variable, which is the pH. So we must have the same pH. And how can we obtain it? By using pH buffer, which is used to obtain a specific pH value. Now, we must write down that we must leave each for 3 minutes by using stopwatch. And of course, we must measure the volume of oxygen produced, which is the most important thing, which is the dependent variable. So measure the volume of oxygen produced by a gas syringe. So we're going to use a gas syringe. And of course, we must talk about safety and a repetition of the investigation. So we're going to write down to wear gloves. We can write down goggles, lab coats as an example. And of course, finally, we must write down to repeat 3 times to take an average. Now we're done with question D. Let's go to question E. Potatoes contains lots of starch. Now, when potatoes start to grow to form new plants, the starch is broken down by the enzyme amylase to form reducing sugars. Now, I must say that amylase is an enzyme that is used to break down starch to form reducing sugars, which are carbohydrates. Now, explain how you could test samples of potatoes to show that starch and reducing sugars are present. Now, we're going to talk about each investigation by itself. Starch, we must add iodine. Secondly, I must say that the color change is going to be from yellow or yellow-brown, let's say, to blue-black. Now, the second part is about the reducing sugars, which is the carbohydrates. We must add Benedict reagent. And then we must heat it in a thermostatic controlled water bath. Of course, we must maintain a specific temperature, which is at 80 degrees Celsius. And the color, we must talk about the color change finally. Changes from blue to brick red. In this way, we're done with question E. Let's now move on to the next question. Now in question 2, we have one more investigation. Here, let's read it together. Now question 2, Woodlice are very small animals. The rate of respiration of a woodlouse can be measured by using a simple respirometer as shown in figure 2.1. Now, as woodlouse respires, the drop of colored liquid moves along the capillary tube as shown in figure 2.1. Now, let's go to the question. Here it says an AI record the position of the drop of colored liquid in the capillary tube shown in figure 2.1 at the start and after 3 minutes. So here we have the start and here we have after 30 minutes. Start, let's use our ruler to see approximately where the line is going to intercept on the ruler. So 2, 3, 4, 5. So this is going to be 4.6 centimeters, or yes, 4.6 centimeters. And right here, it's going to be approximately 0.4 centimeters. So we're going to write down here 4.6 centimeters. We can even convert it into millimeters. So 46 millimeters because of the units right here, minus right here we have 4 millimeters. So it's going to be at start 46 millimeters. And it's already written, so I don't need to write it down. And at the end, it's going to be 4 millimeters. Now, in question II, using the information provided in 2AI, calculate the rate of movement of the drop of colored liquid in millimeter per minute. This is very important because this is the unit used down here. Give your answer to 1 decimal place. So what we're going to do is to divide the difference divided by 30 minutes. The difference that we have, so it's 46 minus 4 because it's talking about the rate of movement. So it moved approximately 42 millimeters in 30 minutes. So I'm going to use the calculator to calculate it. 42 divided by 30 minutes equals to 1.4 millimeter per minute. Now, we're going to write it down. So if you're written down 1.4, 1.3, both are correct. Let's go to the next question. Question B, it says, the rate of movement of the drop of colored liquid along the respirometer can be used as a measure of the rate of respiration. Now, a student used the apparatus shown in figure 2.1, which is the figure in the page that we already solved, to investigate the rate of respiration in different species of small animals. Now, the results are shown in table 2.1 as we see right here. Now, it seems like we're going to plot some data on a graph. Now, in question I, plot a bar chart on the grid to show the average rate of movement of the drop of colored liquid in the capillary tube for the 4 different species of animals. Now, this is the graph given. Now, right here, it says a bar graph. So it's a bar chart to show the average rate. Now, what we're going to do is to take the average, and I wanted to say that after taking the average, we must know that this is the dependent variable. And The animal species is the independent variable, which is very important to know while writing or plotting a bar chart. Now, let's go back. So, of course, we must know that the independent variable is always on the x-axis. So, we're going to write down animal species and here's the y-axis. Now, on the y-axis, we always have the dependent variable. So, what we're going to write down, it's rate of movement of drop colored liquid. Now we must write down the unit because it's very important. They would deduct marks if it's not written down per minute. Okay, now we're going to plot down the animal species. We have 4 animal species, A, B, C, and D. So we're going to go down, plot it right here. So you must have equal intervals. So now I'm taking some intervals. They must be equal. Here I'm going to have species A, here I'm going to have species B, here species C, and here species D. Now before plotting the graph, of course, we must write down the numbers on the y-axis. Now the numbers that we have are between 0.9 and 2.5, so it's not such a large scale. So the scale that I'm going to use is that each large square is going to be 0.5, 1, 1.5, 2, and 2.5. The first one, species A, the average is 1.5. So of course we need to use a ruler. Do it like that. This is the first bar chart. Now we must shade it a little bit so it could be clearly shown on the graph. This is number 1. Now let's go to B. B, the average is 0.9, so that's smaller, and it's species B. So let's go to 0.9. It's going to be approximately right until here. So we're going to put down the ruler like this and shade it again. Species C, the average movement is 2.5, so we're going to take it all the way to 2.5 like this. And we're going to shade it like this. Now we have the last species, which is species D. Right here it says the average is 1.9, so it's going to be approximately until this point. So I'm going to draw a little line right here, shade it down. Now, next question, question double I. State which letter represents the animal species with the higher rate of respiration. It's obviously species C because it has the largest average rate of movement. So we're going to write down species C. Let's go to the next question. In question triple I, the student decided it would be better to calculate the rate of respiration per gram of animals so that values could be compared. Now, the question says, describe how the student could find out the rate of respiration per gram. That's a good question. So it's asking, how can we get the rate of respiration per gram? Basically by measuring the bubble movement. So we must measure the bubble movement. Measure bubble movement. And we're going to divide it, so it's going to be over time. All of this is going to be divided by mass to get it per gram. Now, the next question. Figure 2.2 shows a photograph of a woodlouse. Now, in question I, it says draw a large diagram of the woodlouse in figure 2.2. Now, there are many important things to be stated about this figure, that we should use a pencil to draw, and the picture or the drawing should be at least taking half of the space of the paper provided, and no artistic lines are allowed. So it's simple. I'm going to take the picture itself. You don't have to be an artist to do it. Just a few lines like that. And then you're going to do it like this. I'm going to count the number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. Do it like that, and then we must make the limbs right here. So it's like this, and then like that. Do the other limb. In the picture, it's shown like that approximately. And then you're going to do 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, like that. And of course, we must draw the lines that are shown in the drawing. So it's like this. I'm going to connect all the edges together. And we're all done. Now let's go to the next question. Okay, we're all done. And now here we're in question double I. The magnification of the woodlouse in figure 2.2 is times 9. Now measure the width of the woodlouse along line PQ. Include the units. So now when you're going to measure line PQ, you're of course going to use a ruler, and it's better to convert the units into millimeters. Now calculate the actual width of the woodlouse using the formula include the unit. Now it's asking for the width. So now we have a formula, which is I am. It wants the actual width. So it's actual equals image divided by magnification. Now the magnification, it's times 9. And the image, it's 46 millimeters, as I said earlier. So now we're going to divide 46 divided by 9 equals to 5.1 recurring. Now, it's approximately the same. It depends on the length shown on the drawing you have. So, you're going to get the answer in millimeters. I hope this video finds you well.